My name is Delany Rodriguez. I'm a researcher here at the University of California, Santa Barbara. And we particularly work with ascidians, colonial ascidians in this lab. I've been working here for since 2010, and we were trying to really expand this new model system. What we do is a variety of, we use this particular animal which has a very interesting biology, and what we do is we really use it because it's unique, and we can study a variety of different biological questions. Specifically, what I work on is on vascular biology, and the reason why I study vascular biology in these animals is because these guys have what is called an extracorporeal vasculature, which means that their blood vessels literally come outside of their bodies and extend out. These blood vessels are transparent, they're easy to manipulate, they're easy to visualize. We can do a lot of experimentation with them, and on top of that, if we cut them, they can regenerate. So we can study vascular regeneration, we can study aging, we can study these guys through time, and that's just one of the focus of them. We can study other questions such as non-self recognition. We can study stem cell biology, and this is just one of the many different types of research that we can do in this lab, or we focus on this lab. So I am very interested about the aging process. And part of the aging process, to understanding the aging process, is super important to study the vasculature, because if you think about it, vasculature is essentially like the highway that will deliver the nutrients to all the important tissues and organs, right? And once that highway starts actually going to wear and tear, then you don't get all the nutrients, it's not as effective, and then there's a decline in the overall organism. So just like that happens also in ascidians, and we can visualize this very clearly. And so one of the things that we've noticed is how the vasculature changes through time, how the vascular cells change in shape, and how this has repercussions with the overall physiology of the animals. One of the cool things that we can do here is something called parabiosis, which parabiosis means that you can actually fuse blood vessels from different individuals together. And now what happens is that those blood vessels will start actually sharing blood. So what we do in the lab, we use two different genotypes of these ascidians, we let them fuse, and what is actually very nice about these fusions is that we can do what is called an age mismatch or heterochronic parabiosis, which means we can actually fuse a young animal to an old animal and actually see the effects of what happens when you have young blood going into an old animal, and vice versa. What is in the old blood that makes the young animal kind of starting to behave like an older animal, and the old animal actually behaving a bit more like young. And so we're very interested in understanding what happens to the vascular cells and also to the blood. What is in the blood that gets older that can be reverted, or what is in the young blood that will help to revert the effects of aging. So that's one of the main things that I study in this organism. Older animals, they do start looking a bit younger. We have developed an assay where we actually can induce what we call a vascular regression. So all the blood vessels are outside of the bodies. We can use drugs that actually make them to regress. And one thing that we've noticed is that this is very age dependent. Older animals, if you like, they have stiffer blood vessels that they don't really regress much. Young animals regress a lot. So when we do these fusions of young and old, the older counterpart now actually can regress their blood vessels a lot more, whereas the young animals, they regress less. So there's definitely, we can see the effects of both of them influence each other. There's several things that we can do. We can actually then just cut off the fusion, and one of the things that we're doing now is to look at how long will these animals now can extend their lifespan. Some of the benefits, if you like, for the younger animals is that we also see that they're very young, and we have an old animal that has gonads, then that induce a premature gonadogenic start on the young animals. So essentially they become fertile a lot sooner than if they are not fused. So we're studying all of these different sort of like induced fertility in the young and sort of like this sort of rejuvenation in the older animals.
Cool.
We use a lot of the products from WPI. Specifically, we use a lot of microinjections. We microinject a lot of different dyes into this vasculature that I say it's outside of the body. It's easy to visualize, it's easy to manipulate, and it's actually easy to inject into. So one of the great advantages that we have is that using WPI microinjectors and needles, we can actually go inside of these blood vessels, deliver different kinds of dyes or drugs, and see the effects that we can follow. For example, we can add some specific dyes that will allow us to see the periphery of the cell, so we can actually have like a true, real idea where the cells are in the vasculature and how they're changing through time and what happens when they fuse and all of these different effects that I talked about earlier. Well, my ultimate research goal will be to truly fully understand these aging process, and hopefully to really understand how can we revert it to an extent. So the idea on aging research is perhaps one of the goals is, yes, expand our lifespan as humans. If we can understand it in other organisms, can we apply it to humans? But in reality, one of the things that we in the aging field we're really thinking about is not only trying to live 100 or 120 years old, but really is to expand the health span. So let's say if we're going to live up to 90 years, let's have really good 85 years.
Yeah. Yeah.
So we're trying to understand that that's the reason that will be the ultimate goal of our research, if we can really try to really improve the health span of individuals. When I started in this lab, I started working with stem cells, and I was not working in the vasculature, but for different reasons in the project, I have to keep animals that were getting older and older and older, and I started noticing how they were actually changing physically, having these changes on them. And I thought, okay, no one is really kind of studying this, and I knew that people have published about parabiosis on these animals, and I thought, well, this is a great model that is not being truly used as its potential in this particular area. So I decided to switch fields from stem cells and really try to study the aging process. So I have transitioned into this new field, which is very exciting.
Yeah.